I'm going to talk about conservation of protein structure. There's a connection between protein structure and function. If you change the structure of a protein, you're likely to impact its function either positively or negatively. So proteins evolve. That's how their structures change. Mutations cause changes to genes. Changes to genes lead to changes to proteins, and that changes their functions. Factors that can cause mutations include UV light, which damage DNA, the carcinogens in cigarette smoke, as well as mistakes during cell division. So every day your DNA takes millions of hits, and every day millions of cells die. Both of these cause mutations to occur. When new cells are created, base pairs are copied. So we have changes to protein structure, and they may make no difference. They may improve their function. They may impair their function depending on where in the protein the change occurs, and also what the change is, from which amino acid to which amino acid. Over long periods of time, mutations that confer a survival advantage are incorporated into the gene pool, and mutations that confer a disadvantage are lost. And that's because anybody carrying a disadvantage is less likely to survive. So for a mutation to be passed on from parent to offspring, it has to occur in a germ cell. So that's an egg or a sperm. If it occurs in any other cell, it's not passed on. So this evolution of protein has led to the concept of protein families. So members of the same protein family share common evolutionary history and some sequel and functional aspects. There are databases online that has data on proteins. PFAM maintains a database of protein families. UniProt maintains a database of protein sequences, and the PDB-E maintains a database of protein 3D structures. I know them. I worked with them for 9 good years. So this here, we've seen this slide before, is the structure of insulin. This was covered in lecture 1. And we're going to look at the conservation of the structure of insulin. Okay, what we see here is how insulin is synthesized in the beta cells of the pancreas. It's in the form of preproinsulin. And preproinsulin is cleaved by an enzyme at A24. That's right at the end of the green section to form proinsulin. So the green section is cleaved off. We then have cleaving at 54 and 89. So this means the orange section is cleaved off. So we're left with the red section, which is the B chain, and the blue section with the A chain, and we've lost the C chain in orange and the green chain. And note the black circles. Those depict insulin gene mutations that cause permanent neonatal diabetes. So this shows the importance of these locations, right? Because if the amino acids in these positions change, so wherever we see a change, that mutation stops insulin from doing its normal function and causes neonatal diabetes, permanent neonatal diabetes. So as far as the family is concerned, insulin belongs to an ancient superfamily of functionally diverse proteins. So the function that it carries out today is not necessarily what it would have carried out right at the start in an insect, for example. So insulin belongs to the superfamily, and it's been found in eukaryotes. So that's going back really, really far, almost right to the beginning. And it's found in species such as insects and turtles. So despite broad functional divergence in families, so despite the fact that the protein is doing different things in different species, all insulin proteins, all members of the family, are characterized by 3 domains comprised of the B chain joined to an A chain by a C chain. And the B and A chain are relatively invariant. They don't change much, but the rest of the molecule, they change. So we'll have a look at that. So this is, we're just looking here at the structure of the A chain. And what I've shown there is the insulin signature. So what that means really is that this part is highly conserved, this part is highly conserved, and this part is highly conserved. And these vary as you go from the earliest members of the insulin family to the latest members. And note that these are all cysteine residues, and they're all involved in the disulfide bridges. So the disulfide bridges of insulin are very, very important, and they date right back almost to the beginning of insulin. Now, this is an interesting slide here, because what I've done, I've dug this information out of UniProt, and we can see the conservation of structure of insulin across 3 different species, right? This is the insulin signature. So this part here is the insulin signature. You can see the characteristic C amino acid, cysteine amino acid. You can see that humans do, all these species, human insulin, green sea turtle insulin, and the Anopheles mosquito, which has a insulin-like peptide, all of these comply with this signature, the insulin signature. And interestingly, the human and the green sea turtle, this portion of the insulin molecule, it hasn't changed much really. There's this change here and this change here. But otherwise, the rest of it is the same. So we see the conserved cysteine motifs that characterize the insulin family. It's conserved across these 3 species, which span a wide range of evolution. So what I've done here now, I've compared the structure of insulin to some various species. This is the comparing of insulin to human insulin to gorilla insulin. It's exactly the same, 100% the same. 110 amino acids for both species, exactly the same. This one here, the top row is human insulin, and the bottom row is the gorilla insulin. The B chain, exactly the same. The C chain, the A chain, exactly the same as is the C chain. It's quite remarkable because man and gorilla last shared a common ancestor about 8 million years ago. The picture is quite similar for the chimpanzee. The difference is, there are differences here and here. And those are not in the important parts of the insulin molecule. The B chain is unchanged and the A chain is unchanged. This is pig insulin. We last shared a common ancestor with pigs between 79 and 97 million years ago. And what we see here is that we're beginning to see now more variation. Look at there's a lot of mutations here in the C chain and quite a lot in the other part of the molecule, but the B chain only has one difference. And that's even though it's like 70 million years or so ago when we last shared the common ancestor. Okay, mouse shows a similar story as pigs, and we diverged, mouse and man, between 65 to 75 million years ago. Now, I tried to find a molecule as far back as I could go, and I did manage to dig out the green sea turtle. And I'm not sure how long ago we diverged, but it's probably more than 100 million years. These turtles have been around for a long time. And we can see that there's not a lot of differences between the B and the A chain, but there are lots of differences in the C chain and also the other part of the molecule, even though we're quite distant relatives. So this summarizes the how we're linked to our nearest relatives, nearest living relatives. And we see that our nearest relatives really is the chimpanzee, because we diverged here, which is about 6 million years ago. And we diverged from gorillas about 8 million years ago. and we diverged from orangutans about 13 million years ago. And all of us here have pretty much the same structure for insulin A and B chains. Okay, so I'm going to talk about hemoglobin now to illustrate a slightly different concept, but it's to do with conservation of structure. Normal hemoglobin is made up of 2 alpha chains and 2 beta chains as shown here, and sickle cell anemia is caused by a mutation in the beta chain. So here, this is normal, the beta chain of hemoglobin. This is the mutant beta chain of hemoglobin, and this is the mutation here. Just one substitution really, where T is replaced by A. The result of that is glutamate is replaced by valine in the protein, and that causes problems. The effect of that really is, instead of having glutamate, which has a negative charge, we have valine, which has no charge. So this likes water, and this doesn't like water. This is hydrophobic, valine. So we replace a negative charge here, and so there's no negative charge. So we have a sticky hydrophobic part here and here, as glutamate is changed to valine. And what that does is it causes clumping of the proteins. So under normal oxygen, the mutation causes no apparent effect on the secondary, tertiary, or quaternary structure of hemoglobin. So we don't even notice it under normal conditions. But under low oxygen, the hydrophobic valine creates this sticky patch I was talking about, the hydrophobic patch, which causes the hemoglobin molecules to aggregate to form fibrous precipitate. So it comes out of solution and it's a solid. And that distorts the red blood cell to form the sickle shape. And that has some quite serious consequences. For one, the sickle cells die within 10 to 20 days as opposed to 120 days for normal cells. And that may create a deficiency in red blood cells if your body is not able to replace the dead cells. It has to work faster. And the sickle cell shape blocks blood flow to limbs and organs, so that may cause a range of symptoms from mild fatigue to organ damage to severe pains, and also increased risk of infection. So if sickle cell is so bad and it gives you a disadvantage, why does sickle cell anemia, why is it still around? Well, there's a connection between sickle cell and malaria. The graph there shows the distribution of sickle cell anemia. You'll see it's common in warm countries where you're likely to find mosquitoes that spread malaria. And if we look at this map here, it shows on the left sickle cell, the presence of the sickle cell allele in Africa, and compare that to the graph on the right, which shows the existence of malaria. You see there's some correlation there. Sufferers of sickle cell, the full sufferer, to get sickle cell anemia, you have to have both genes defective. And people who have both genes defective, they are not protected against malaria. But what happens if you have one copy of the faulty gene, right? You have what is called a sickle cell trait. And if you have that, you're 60% less likely to die from malaria than someone who has normal blood. And so you tend to get a higher frequency of the sickle cell genes in area where malaria is common. So a sickle cell trait protects against malaria, so it gives you a survival advantage, which is why this mutation exists. In areas where malaria is common, 10 to 40% of the population carry this trait. So it shows preservation of a mutation that provides a survival advantage. So just to summarize, amino acid sequences of proteins are required to serve important functions and are conserved over millions or even hundreds of millions of years. Right? So those part of the protein that serve important functions are conserved, and those part of the proteins that provide a survival advantage, they are also conserved. And that's it.
Marcus Garvey words come to pass. Marcus Garvey words come